Hello everyone. Today we are going to discuss the pancreatic islets. In this picture, according to the organ's location and morphology, we can easily identify the pancreas. The pancreas is a mixed exocrine-endocrine gland. The pancreatic islets compose the endocrine pancreas. The reason why we call it islet is that isolated island-like cells are dispersed in the exocrine pancreas. There are more than 1 billion islets in pancreas and mostly in the tail region. These islets can produce secretions, but they have no duct, so they belong to the endocrine system. After this session, you should be able to describe the structure of islets, list their homes, and explain the associated clinical changes with pancreatic islet. Talking about the structure and function of pancreatic islets, we will start from two young scholars. One is the German pathologist Paul Langhans. He was born in Germany in 1847 and studied medicine at the University of Jena and Berlin. In 1869, 22-year-old Langhans detailed the microstructure of the pancreatic islet for the first time in his graduation essay. He described several different types of cells, including irregularly shaped polygonal small cells. These happen in the entire gland, which literally means a cell stack in a diameter of 0.1 to 0.24 millimeter. This is what we call pancreatic islet today. As we can see in the picture, they are lightly stained cell clusters and scattered among the exocrine pancreas. It was Paul Langhans who firstly discovered and described this structure. So, in 1893, the French histologist G.E. Langhans named these structures islets of Langhans. Langhans was just a medical student at 22. He discovered a new structure of human body through careful observation. There are still many mysteries in the human body, so you also have chances to explore them during your study. However, Paul Langhans did not make any assumptions about the function of the pancreatic islet and the nature of the cells. The discovery of important functions of the islets was by another young scholar, Frederick Benton. Benton was born in Ontario, Canada, in 1891. He studied as an art student at the University of Toronto, but later switched his major to medicine. After graduation in 1916, he joined the Canadian Army Medical Corps and served in France during the First World War. When the war ended, Benton returned to Canada and tried to be a medical practitioner, but there were not enough patients for him to carry on. Eventually, he did part-time teaching at the University of Western Ontario. In 1920, 29-year-old Frederick Benton was preparing an experiment on pancreas function and glucose metabolism. He found that from the late 19th century to the early 20th century, many scholars speculate that diabetes was related to pancreatic hormone. They call this hormone insulin. However, eating animal pancreas didn't help in treating diabetes. Considering the functions of the digestive tract. He suspected that eating animal pancreas may have caused its home to be destroyed by proteases in the GI tract. Meanwhile, a surgical journal article mentioned that pancreatic tumors could compress the secretion ducts and cause atrophy of the exocrine gland. He was greatly inspired and made the following animal experiment plan. He would ligate the pancreatic ducts to make proteases-producing cells atrophy without affecting insulin-producing cells. Then, injecting the pancreas extract could lower blood sugar in diabetes. Working in Professor McLeod's lab in Toronto, although Banting and another collaborator named Best failed many times, they gradually improved the experiment. They ligated the pancreatic ducts in dogs and got the pancreatic extract. Then they injected it into the experiment diabetes once. This experiment finally proved that pancreatic extracts contain the function of lowering blood sugar and treating diabetes. After their success, insulin was purified and put into clinical use not long after. In 1923, Banting and McLeod were awarded a Nobel Prize in Physiology and Medicine for their contribution to the discovery of insulin. Banting raised the question at 29 years old and won the Nobel Prize 3 years later. Their success was the result of persistence and resilience. But pancreatic islet is not just composed of one type of cells. It can do more than secreting insulin. The conventional hematoxylin and eosin staining could not distinguish various cells in the pancreas. With special staining, we will find that there are many types of cells in the pancreatic islet. Among them, the three main types are A cells, B cells, and D cells. A cells surrounding the pancreatic islets and account for about 20% of the total cells in the islets. They produce glucagon to increase blood glucose by accelerating hepatic glucogen degradation and gluconeogenesis. B cells locate in the center of the islet and account for 70% of the total cells. The substance produced by B cells is insulin. The main function of insulin is to decrease the blood glucose by causing glucose entry into body cells. If the activity of B cells is decreased, for example, destroyed by pathological autoimmune response, the insulin will decrease and the blood glucose cannot enter cells. This condition will lead to high blood sugar but insufficient glucose for cellular function. Eventually, it can cause the clinical manifestation of diabetes. The third main type of the cells are called D cells, which make up about 5% of the total pancreatic cells. They are scattered between A cell and B cell and produce somatostatin. D cells are paracrine cells, so its harms can affect the adjacent cells. That is, somatostatin could regulate the secretion of A cells and B cells. Above is the introduction about the pancreatic islet. Now, do you understand the work of the two young scholars associated with our pancreatic islet? Langhans identified the pancreatic islet in morphology. Banting discovered the role of insulin. Also, the most important function of pancreatic islet. For you, my young medical students, please summarize the structural features and functions of pancreatic islets based on this picture. Here are the references. Thank you.